Hey, hello guys, good evening to everyone and welcome back to IQCIVILS IAS Academy after a long break. Like I guess I haven't uploaded any videos since last 2 weeks on our IQCIVILS IAS Academy channel, but from today onward I will be regular with you either 3 times a week or maybe every day. Okay, fine. So today, welcome to IQ Civil Science Academy, a great coach transforms life. Today we are going to discuss one important topic called gene therapy. We actually already discussed about gene therapy under one video called CRISPR-Cas9. So what is that there was a recent news that is related to the gene therapy, and while discussing that news we will also discuss about the CRISPR-Cas9 and what exactly gene therapy is. This topic will be covered by me, Dr. Sachin Kadam, faculty at IQ Civils IAS Academy, Guwahati. This is about IQ Civils IAS Academy. You can see ours is an academy which is a government affiliated academy. Then we went through different projects in collaboration with Assam Administrative Staff College since 2018 till date, and even this time if you went through the result of APSC, IQCIVILS IAS Academy helped many students to crack APSC examination. This is the Google review given by the students to IQCIVILS IAS Academy, 4.9 star, that is rare and unique for a civil services institute. This is our elite panel of faculties from Delhi that includes Sunil Jha sir, Sachin sir, Anuj sir, Alok sir, Yogesh sir, Rizwan sir, Vikram Singh sir and Nitin Kumar sir. Now guys, we are starting our new batches from coming Monday, that is 14th of November. We already launched one batch on 10th of November and second batch will be launched on 20 November. If you want to book your seat and if you want to register yourself for the admission at IQ Civil Science Academy, do contact Anuj Singh sir, who is the director of IQ Civil Science Academy. Number is given, or you can connect to Vishal Sharma, our admissions manager, or you can connect to Anuradha Saikia. This is our YouTube channel. You should subscribe to our YouTube channel to get all the latest update related to the APSC and UPSC. And don't forget to join our 10th or 20 November batch for UPSC and APSC examination. So what was the news exactly? So news was death in US gene therapy study, an ethical issue. Now see, if you observe the concept of gene therapy, this itself is an ethical issue, and use of gene therapy in humans is actually not allowed. It can be allowed either for a research purpose or in cases of exemption where there is no other treatment to treat a particular disease in a human. So there courts can allow to use gene therapy. For example, your UK court allowed mitochondrial gene therapy or three parents baby under this type of exemption. So यूएसए में जीन थेरेपी की ट्रीटमेंट की जा रही थी, तो उसमें द पर्सन हु वाज इन्वॉल्व, द वॉलंटियर, ही डाइड. तो व्हाट वाज द न्यूज़? So death in US gene therapy study, a lone volunteer in a unique study involving a gene editing technique has died. So there was a single person who was volunteering so that we can study the role or we can study the importance of gene editing technique or how exactly it works in human, and that volunteer died. And it has raised the concerns regarding gene editing technology. Now see, when you are testing gene editing technology on human, and if that is leading to the death of human, so it is an ethical issue, right? So that is the reason why gene therapy came into current affairs or came into news. What is exactly the issue was, Terry Horgan, who was a 27-year-old person, he died last month, and he was under trial with a gene editing tool called CRISPR-Cas9. So here you need to remember that the first point, if you want to edit genes of any living organism, so you can use one tool. The name of that tool is CRISPR. The first point, CRISPR Cas9 is a gene editing tool. This is the first point and you need to remember this. Okay. So Terry Horgan, a 27-year-old, died last month. He was under trial with a gene editing tool called CRISPR to treat a Duchenne muscular dystrophy, the rare genetic muscle wasting disease. Now we will check out what exactly this Duchenne uh, muscular dystrophy. This is the disease. What exactly happens in this disease? So in this disease, the person slowly loses the muscle mass. Person slowly loses the muscular capacity. So here you can see progressive weakness and wasting of muscles. So muscles of that person will start slowly, they will start destroying, they will start weakening. Onset 3 to 5 years old, genetic, primarily genetic reason है, and very important this disease is mainly found in male. Females may भी हो सकता है, but you can find this mainly in male. History of motor development delay, clumsiness, frequent falls, difficulty climbing stairs, running and riding tricycle. Waddling gait like this. This is how it happens. Okay. Ambulation frequently impossible by age 12. Breathing muscles become more affected, and for your information, a person with Duchenne's muscular dystrophy either dies because of failure of respiratory system or because of failure of heart function. Our respiratory system works with the help of muscles and our heart itself is made up of muscles. So death is either because of heart failure or because of the respiratory failure. As breathing muscles become more affected, life-threatening infections are common. This usually leads to death by age of 15 to 18 years. Okay, so this is Duchenne's muscular dystrophy, or you can check it out. We will see some more examples. So here you can see decreased heart function, cardiomyopathy means death of the muscles of heart, heart failure, weak diaphragm, respiratory failure, and loss of muscle mass, inflammation and fibrosis. It requires wheelchair. So in this disease, actually a person with this disease loses the muscles because of genetic disorder, and that disease is called Duchenne's muscular dystrophy. It is caused by a mutation in the gene which is responsible for the production of dystrophin protein. That protein is not formed by that gene, so that is a mutated gene, and because of this mutated gene, this Duchenne's muscular dystrophy happens. So Terry Horgan, who was 27 years old, he was infected with a genetic disorder called Duchenne muscular dystrophy, and to treat this genetic disorder, scientists in USA were using CRISPR-Cas9 technique, and while doing so, that patient died. So that raised some ethical issues ki whether we should use CRISPR-Cas9 or whether we should use CRISPR-Cas9 gene editing technology or gene editing technique in the treatment of genetic disorders. Okay, most people with Duchenne die from lung or heart diseases caused by it. Now for your preliminary examination, they can ask you कि Duchenne disease is related to, so it is related to muscular dystrophy. Okay, it is in news, so for sure definitely they can ask you a question about Duchenne muscular dystrophy. So because of this news, gene therapy came into news or current affairs. Now what is gene editing? We will study this gene editing in detail. It is a type of genetic engineering in which DNA is inserted, deleted or replaced. So this is DNA. Either you will insert new DNA or you will delete part of the DNA, or you will remove one part of the DNA and replace that with another part of the DNA. So DNA inserted, deleted or replaced in the genome of an organism using artificially engineered nuclease or molecular scissor. Now what is the meaning of molecular scissor? These are the molecules or enzymes which are used to cut DNA molecule. So we call them molecular scissor. And these molecular scissors in gene editing technology or in CRISPR-Cas9, they are artificially produced, artificially manufactured, artificially engineered. So when you use artificially engineered molecular scissors to insert, delete or replace parts of DNA or parts of a genome, that technique or that method is known as gene editing. And most commonly used gene editing technology or method is CRISPR-Cas9. Okay. So we will come back to the ethical principles during clinical trial, but before that, what is DNA? The long form of DNA is deoxyribonucleic acid. DNA is responsible for carrying information from one generation to another generation. So we call this hereditary material. We also call DNA genetic material, or we also call DNA storehouse of the genetic information. Nearly every cell in a person's body has the same DNA. See, whether it is a skin cell, heart cell, brain cell, liver cell, any muscle cell, any cell in our body has the same DNA. Only DNA is not present in RBC. RBC is a cell, but you will not find DNA in RBC, or DNA is also not present in the platelets. So these are the cells present in the blood where you will not find DNA, but in every other cell in human body you can find DNA, and that DNA is hereditary material, that DNA is genetic material, that DNA is storehouse of genetic information. Most DNA is located in the cell nucleus, but in some cases DNA can be located in the mitochondria. So mitochondria is a cell organelle, so DNA can be found in mitochondria. DNA can be found in nucleus. Most DNA is located in the cell nucleus, but a small amount of DNA can also be found in the mitochondria, as I already told you. So you need to remember that DNA can be found in nucleus or it can be found in mitochondria. Okay. Next, now see, genome means all the chromosomes that are present in the nucleus of a cell. We call that genome. Now these chromosomes are made up of DNA, and this DNA contains genes. So more than one genes are present on the DNA, and these genes are responsible for the production of different types of proteins. So DNA contains genes, and all the genes present on DNA, all the genes present on a single DNA, we call them genome. What we call them? We call them genome. So what is the meaning of genome? It is all the genes present on the DNA of an individual or of a single individual. And when you pack this DNA so that information can be transferred from one cell to another cell in a packaged form, we call that chromosome. Clear? So what is DNA, what is chromosome, what is gene, and what is genome? Now sickle cell anemia, it is a genetic disorder in which the normal shape of the RBC is like this, but because of genetic disorder, defect in the genes that are responsible for the formation of RBCs become sickle shaped, and that leads to anemia. So we call that sickle cell anemia. Okay, let's go ahead. So what is CRISPR-Cas9 system? To see, first thing, a death of a patient in USA during gene therapy trials or gene therapy study. That person had the disease called Duchenne muscular dystrophy, and to treat this disease, we used one gene editing tool called CRISPR-Cas9. Now, what is CRISPR-Cas9? So, over last 3 years, gene editing technology with nearly unlimited potential has produced flawless results in clinical trials. India has approved a 5-year project to develop CRISPR to cure sickle cell anemia. Very important to remember. India has approved a project in which we are planning to use CRISPR-Cas9 technique to treat sickle cell anemia. Okay, now what is CRISPR-Cas9 technique? Genome editing, or also known as gene editing, it is a technology that allows genetic material to be added, removed, altered, or deleted or replaced at a specific location, not everywhere. The part of the DNA which contains a defective gene, only that gene will be altered, only that gene will be removed, or only that gene will be replaced, and rest part of the DNA remains as it is. So the unique thing about CRISPR-Cas9 is that it is very accurate. When you use CRISPR-Cas9 technology, so this CRISPR-Cas9 technology never lead to the problem of mutations in other parts of the DNA. That's why we say that CRISPR-Cas9 technique is very, very accurate. Okay. Now working on CRISPR, its mechanism is often compared to the cut, paste and cut, copy, paste or find, replace functionalities in common computer program. Yes, this is DNA. So if this part of DNA is defective or this part of DNA is not working really well, so this part will either be removed or this part will either be replaced by another better DNA, and for that you need to cut this DNA, then you need to copy healthy gene and paste it here. So cut, copy, paste, or find the defective sequence of DNA and replace it with healthy sequence of DNA. That we call cut, copy, paste or find, replace functionalities in common computer programs. A bad stretch in DNA sequence, which is the cause of disease or disorder, is located, cut and removed and then replaced with a correct sequence. That's why we call this cut, copy, paste or find, replace functionalities. Okay, so this is how CRISPR-Cas9 works. Now DNA editing, what exactly or how exactly we do DNA editing by using CRISPR-Cas9? A DNA editing technique called CRISPR-Cas9 works like a biological version of a word processing program's find and replace function. How the technique works? So this is the cell. Inside cell there is a nucleus, and inside a dividing cell's nucleus chromosomes are present, and these chromosomes are made up of DNA. DNA is made up of genes. So we identified a particular stretch of the chromosome or DNA. Okay, just give me a minute. Particular stretch of the DNA which is defective. So see, a cell is transfected with an enzyme complex containing guide molecule or guide RNA, healthy DNA copy or healthy gene, and DNA cutting enzyme called Cas9 protein. So a specially designed synthetic guide molecule or guide RNA finds the target DNA strand. So this is the target DNA strand that will be found by the guide molecule or guide RNA. Then with the help of this guide molecule or guide RNA, DNA cutting enzyme called Cas9 comes to the place where there is a defective gene. It will cut that defective gene from the DNA, and और क्या था हमारे पास healthy DNA copy. This healthy DNA copy will be inserted. So cut and paste, या cut, copy and paste. That is what we did by using number 1 guide molecule, number 2 healthy DNA copy, and number 3 DNA cutting enzyme. This is how CRISPR-Cas9 works. So CRISPR-Cas9 is a unique technology that enables editing parts of the genome by removing, adding or altering sections of DNA. Guide RNA guides Cas9 to the right part of the genome. Enzyme Cas9, it acts as a pair of molecular seizure. So number one, search. A CRISPR molecule finds a precise location in the target DNA. Number two, cut. The CRISPR enzyme cuts the target DNA at the point found by the guide. And edit. A new custom sequence can be added when the DNA is repaired. So this is how CRISPR-Cas9 technique works. Now, what are the potential applications of genome editing technology or gene editing tool? So first, if we can edit genome, so before editing genome, we need to study genome really well, right? Without studying or without finding out right sequences of DNA, we cannot go ahead with CRISPR-Cas9 technique, right? And when you study DNA in a right way, in a perfect way, so it helps you to understand or better understand diseases. So the first application of CRISPR-Cas9 is better understanding of diseases. Most uses of genome editing have been in scientific research, for example, to investigate models of human disease. Number two, several therapeutic interventions using CRISPR for diseases like thalassemia or sickle cell anemia have gone into clinical trials. This will avoid most of the avoidable deaths in poor countries. So first, CRISPR-Cas9 will help us to better understand, to get better understanding of the diseases. Then, CRISPR-Cas9 can also help us to identify threats or identify defective genes present in the genome. So genome editing has been used to develop a suite of tools that scientists can use to better understand new and existing pathogens. This can help to develop and poor countries are prepared for incoming threats of disease. So we can predict diseases in advance if we have CRISPR-Cas9 technology or CRISPR-Cas9 technique. Then it also helps us to develop new treatment. Obviously, without CRISPR-Cas9 technique, can we develop gene editing tools? Can we develop can we do gene therapy? So no. So it helps in development of new treatment. Now I will tell you, there is one treatment called CAR T cell therapy, and this therapy is used in the treatment of cancer. Okay, so developing new treatments, the potential of genome editing to impact the development and use of new treatments is tremendous. Genome editing technologies have a major advantage over traditional drugs in that they can target the genetic basis of disease. This can drastically lower the cost of life-threatening diseases. So first benefit, better understanding of disease. Second benefit, identifying threats. Third benefit, developing new treatments. Fourth benefit, treating genetic diseases. That is what we were doing in today's news, right? We were trying to treat the Duchenne muscular dystrophy disease, but that person died. So it has huge potential in the field of genetic diseases. Poor countries like Nigeria, Sudan, and tribals in India have witnessed the rise in cases of diseases like sickle cell anemia and thalassemia. Other potential uses, genome editing technology also have a number of relevant applications to global health security outside the human body. For example, this gene drive is very important. We will discuss gene drives separately in next video. Okay, so gene drives have the potential to control vectors and minimize the possibility of certain outbreaks whose impact is felt by poor countries. So by using CRISPR-Cas9 technique, gene editing technology, we can develop gene drives, and these gene drives can be used to deal with or to reduce the population of some vectors which cause communicable diseases such as mosquitoes. It can help either by eliminating the vector entirely or by editing the vector's genome to remove its ability to carry a particular agent. Okay, so what is gene drive? यहाँ पे इन्होंने already gene drive के बारे में बताया है, but before we go to the gene drive part, let me explain you. अब देखो पूरा एकदम शॉर्ट में मैं आपको बताता हूँ, एंड देन वी विल कम टू द एथिकल इश्यूज रिलेटेड टू द gene therapy. So one volunteer who was being treated for a genetic disorder called Duchenne muscular dystrophy, he died while treatment. To treat him or to treat his genetic disorder, we used CRISPR-Cas9 technology or CRISPR-Cas9 gene editing tool. Now, what is gene editing? When you insert, delete or replace the genome of an organism using artificially engineered nucleases or molecular scissors. What is DNA? Deoxyribose nucleic acid, which is a hereditary material as well as genetic material, and the same DNA is present throughout the cells of an individual. So CRISPR-Cas9 is nothing but a cut, copy, paste or find replace mechanism. And in CRISPR-Cas9 there are two main components. Number one, guide molecule or guide RNA, and number two, DNA cutting enzyme. And along with that, we also provide healthy gene which will replace the defective gene. And how it happens, I just explained you. Okay, so guide RNA and Cas9 protein. How we can use CRISPR-Cas9 technology or gene editing technology? Number one, better understanding of diseases. Number two, identifying threats. Number three, developing new treatments. Number four, treating genetic diseases. Number five, we can use CRISPR-Cas9 technology or technique for the development of gene drives. Okay. Now, what is gene drive? पहले तो it is a type of genetic engineering technique. Very important. Gene drive itself is a genome editing technology or genetic engineering technique that modifies genes. So in gene drive, we modify genes so that they don't follow the typical rules of hereditary. So what is the main objective of gene drive? The main objective of gene drive is to ensure that a particular gene is inherited to the next generation compared to other genes. So gene drive कर क्या रहा है? It is helping us to change the typical rules of heredity. Gene drives dramatically increase the likelihood that a particular suite of genes or a particular group of genes will be passed on to the next generation. So we can use gene drive to ensure that a particular group of genes get inherited to the next generation. So gene drives dramatically increase the likelihood that a particular suite of genes will be passed on to the next generation, allowing the genes to rapidly spread through a population and override natural selection. So when you use gene drive genetic engineering technique, it helps you to spread a particular group of genes across a population. Thanks to CRISPR-Cas9, the gene editing technology harnessed from bacteria, gene drives are becoming easier for researchers to build. So I can say gene drive are currently being used to treat those diseases which are spread by vectors. Now with gene drive technology, you can modify evolutionary trajectory, yes, because you are changing the pattern of inheritance. Okay, this could be an effective way to eradicate nuisance species such as malaria causing mosquitoes. You can make changes in their genes. So CRISPR-Cas9, why this CRISPR-Cas9 was in news? Because in 2020 Nobel Prize of Chemistry was given to these two women, and they were involved in the research or development of CRISPR-Cas9 system or CRISPR-Cas9 technology. What are the opportunities or benefit? We can use CRISPR-Cas9 technique for targeted medicine. We can use CRISPR-Cas9 technique in biofuels. We can use CRISPR-Cas9 technique in disease-resistant crops, and we can use CRISPR-Cas9 technique in new industrial products. But there are some issues. Number one, safety issue. अभी हमने देखा we used CRISPR-Cas9 technique to treat a particular genetic disorder and that person died. Number 2, ethical concern. We will check out them now. And number 3, regulatory challenges. So these are some challenges related to the CRISPR-Cas9 technology or CRISPR-Cas9 technique. Now, what government of India is doing in the field of CRISPR-Cas9? As I already told you, government of India has approved a 5-year project to treat sickle cell anemia by using CRISPR-Cas9 technology. Then, designer baby, it is an ethical dilemma. In 2018, a Chinese researcher disclosed that he had altered the genes of human embryo to prevent the infection of HIV. Remember, genetic engineering or any changes in the genes of human or cells of human is completely banned across the world or throughout the world. Okay, changes in the embryo pass on to the generations. The problem क्यों हमने, why we see, there are two types of genome editing. Either you can do genome editing in somatic cell. So if you make changes in the genes of my skin cell, they will not be transferred to my next generation. But if you do changes or genetic changes in my germ cells, we call that germline editing, then that is a problem. That will create a problem. Okay, so this germline editing is strictly banned. Somatic cell editing, maybe in rare cases or in exception, it can be allowed. Okay, now before we come to these questions, let me explain you what are the ethical issues or ethical challenges related to the gene therapy and clinical trials. So number 1, when you select a subject, for example, you chose that guy who was a 27-year-old with a genetic disorder. Now अब उसकी कोई गलती नहीं, but he died because of the clinical trial. So first, when you are performing clinical trial for CRISPR-Cas9 technique or technology that can lead to the death of humans, then CRISPR-Cas9 technology, it is a new technology. It is not scientifically proven, and it will not be scientifically proven until and unless we do different experiment like the one we did in this case, but that volunteer died. So scientifically designed trials are must. Third problem is informed consent. Many a time doctors or scientists actually do not prefer to inform patient what exactly they are doing, and without informing patient, without his consent, if you are using some medicine on human, so that is completely unethical. Subject should be informed about risk and benefit. You should tell all the benefits as well as risks of genome editing or CRISPR-Cas9 technique. Safety issue, safety of subject should be priority, but in this case we saw that subject died, so safety of subject is a problem. Then justice and equality, trials should be equally available to every section of the society. Okay, so these are some ethical concern, as well as there is one ethical concern of development of designer baby. There is one ethical concern of your changes in the embryo, they get passed on to the next generation, which is a problem. So check out the question. This was the question asked in prelims 2019 or UPSC CSE prelims 2019. What is the Cas9 protein that is often mentioned in news? अभी मैंने आपको बताया था, a molecular seizure used in targeted gene therapy, a biosensor used in the accurate detection of pathogens in patients, a gene that makes plants pest resistant, and a herbicidal substance synthesized in genetically modified crops. Obviously, it is a molecular scissor used in targeted gene therapy. RNA molecule acts as a guide RNA, and Cas9 protein acts as a cutting mechanism, that is molecular scissor. Check out the second question. This question was asked in UPSC CSE prelims 2017. With reference to agriculture in India, how can the technique of genome sequencing, often seen in the news, be used in the immediate future? So actually they are asking you what are the applications of genome sequencing in agriculture, and you should know. So when you study the biotechnology, you can easily understand what are the different applications of genome sequencing. So normally, genome sequencing can be used to identify genetic markers for disease resistance and drought tolerance in various crop plants. It can also be used or helpful in reducing the time required to develop new varieties of crop plants, and it can be used to decipher the host pathogen relationship in crops. So all these three things can be done by using gene editing technology or genome editing technology. So you can see genome sequencing is a laboratory method that is used to determine the entire genetic makeup of a specific organism or a cell type. So guys, we spoke about what is CRISPR-Cas9 technology, how this CRISPR-Cas9 technology is useful, what are the major challenges in the use of CRISPR-Cas9 technology, why this CRISPR-Cas9 technology was in news in 2022. So we comprehensively covered the topic or concept of CRISPR-Cas9. Okay. So this is all from my side. This is IQ Civils IAS Academy, a great coach transforms life. We are starting our new batches from 20 November. So don't forget to register for our new batches. And if you want to register to our new batches, you should call on these numbers. These numbers are of our director, Mr. Anuj Singh, our admissions manager, Vishal Sharma, and our front desk officer, Anuradha Saikia. So that's it from my side, guys. See you soon. Bye bye. Take care. Sayonara. I'm like waiting to see you again.